Hello, everybody. So I'm going to discuss this question. Question is, inherited mitochondrial diseases affect muscle function. Inherited mitochondrial disease, kind of a genetic disorders in mitochondria. It has a defective ATP production. As a result, muscle function is altered due to lack of ATP. Right now, this is the electron transport chain, which is familiar to us still. I would like to highlight the important point of this electron transport chain. Here, the first complex, complex 1, reduce NADH, release the electrons and proton, and it become oxidized NAD. And this electron moving to complex 1, coenzyme Q, complex 3, cytochrome C, complex 4, and this electron finally combine with the proton and oxygen to form water molecule. FAD, reduce FAD, also release electron and proton, and then it become oxidized NADH. FADH. And this electron again transport to complex 2, coenzyme Q, complex 3, cytochrome C, and complex 4. Again, complex 4, electron join with proton and oxygen to make water molecules. So this is here oxygen act as a final electron acceptor. Okay, by doing that, the energy trapped in this electron used to pump hydrogen ion out of this complex. So when hydrogen ion pump into the intermembrane space, create an electron gradient. As a result, proton come through the ATP synthase complex to produce ATP, oxidative phosphorylation. Right, let's continue the question. And investigation samples of muscle tissue from healthy individuals. So here, if you notice, it's healthy individuals. Healthy individuals. So many samples collected from healthy individuals and 4 individuals with mitochondrial disease. The function of mitochondria when these samples were treated with different substrate was investigated. Treating muscle tissue from healthy individuals with pyruvate and malate, succinate, reduced TMPD. So when we treat these with these treatment, increase production of NAD, FAD, and increase reduction of cytochrome C. So that means more NAD produced. This NAD feed into the complex 1 and oxygen production increase. And more FAD produce, feed into the complex 2, more oxygen produce. And reduced TMPD increase the reduction of cytochrome C. That means more electrons are moving forward and increase oxygen production. Oxygen consumption of the muscle tissue sample from healthy individuals and 4 individuals with mitochondrial disease were recorded. After that, the result compared with the individual 1, 2, 3, and 4. Now here the result, oxygen consumption level is 21 plus or minus 5. Oxygen consumption, succinate complex, succinate treatment 13.5 plus 4.5. Reduced TMPD treatment 63 plus or minus 20. Right, so we can consider this as the mean value and the standard deviation. If we consider this as a mean value and standard deviation, so healthy individuals, we can see this kind of a result. That means the mean value is 21, and if we want to find the highest and lowest value of this standard deviation, the highest value 21 plus or minus 5.0. So lowest value is 21 minus 5.0, 16. Highest value is 21 plus 5, 26. So then we can get the range. The range is 26 and 16. So this is the range, standard range. If a person has a lower range below 16, then oxygen consumption is not normal. There is a defect. So we have to find in all treatment which individual had defective treatment and healthy range in healthy individuals range. Okay, here the question. The result of this investigation can be used to identify defect in the electron transport chain. Comment on the defect in the electron transport chain in the individuals with mitochondrial disease. That means we have to find which person has defect. If this person has a defect, which level we can find the defect. And when we comment, when we conclude like that, we have to also give some kind of explanation and theoretical knowledge. We have to use theoretical knowledge to find this defect in each person. So first we have to identify which person has a defect. Right, we analyze this result now. When we analyze the result, we get the range of each treatment. Now, when we get the range of each treatment, the pyruvate and malate treatment, 16, 26. So below 16, then there's a defect. So 3, 18 is okay, 6.5, 7.5 below 16. Succinate, lowest value is 9. Then there is a defect, okay, okay, defect. Reduced TMPD, below 43, there's a defect. 13, 16 is okay, 60 is okay, 51 is okay, 40 is below 43, 63. So then we have to write the result as a first part of the answer. So individual 1, oxygen consumption has a defect in each treatment. Individual 2, oxygen consumption is normal. Individual 3, defect is in a pyruvate and malate complex, succinate and reduced TMPD. There's no defect of oxygen consumption. And individual 4, again, each treatment, oxygen consumption lower. Right? So this one you can nicely write in Word to start the answer for the question. So no treatment stimulate normal oxygen consumption. Pyruvate, uh, all three treatments stimulate normal oxygen consumption. Succinate and reduced TMPD stimulate normal oxygen consumption. Right, now we try to understand how this treatment work. So let's take individual 1. Individual 1, there's a defect in all three treatment. So we start with the reduced TMPD. So when we start to give reduced TMP to the cytochrome C complex, if you look healthy individual, oxygen consumption increase 63. But here oxygen consumption not increase. So that means if we try to feed like this, it doesn't go from cytochrome C complex forward. The electron flow does not take place from cytochrome C onward. So if we feed complex 2, again it doesn't move forward, and it can maybe move up to cytochrome C. And complex 1 again start to move forward, but cytochrome C stuck. It's not moving from cytochrome C. That means there is a defect either in cytochrome C or complex 4. So this point forward, individual 1 has a defect. So individual 2, pyruvate and malate treatment, complex 1, the flow is moving forward and is in normal range. Complex 2, moving forward and normal oxygen consumption, we can see. Cytochrome C, same, moving forward and normal oxygen consumption is there. That means it will these complexes from this point onward, the electron transport chain is working and there's no defect. That means defect is before complex 1. We can assume it should be before complex 1. It could be a other kind of problem, could be Krebs cycle, could be link reaction, or any other defect before complex 1 we can identify. So we go to the individual 3. Individual 3, succinate treatment and reduced TMPD, okay. So then he has a problem with pyruvate and malate treatment. So when we feed pyruvate and malate, more NAD, then complex 1, it doesn't move forward. Complex 2, it's moving forward. Cytochrome C, there's no issue, it's moving forward. That means defect is complex 1. Individual 3, defect is in complex 1. Individual 4, all treatment, oxygen consumption reduced. Complex 1, complex 2, complex 3. So that means, like first one, this is the problem in cytochrome C, because if we test cytochrome C only, it doesn't work. That means complex 1 and complex 2, these should be okay. So defect in cytochrome C or complex 4. From this point onward, there is a defect. Right, now we have to write this part as a second part of the answer to get the full marks. So we can write when we treat with pyruvate and malate, it increase NAD. This reduce NAD convert into oxidized NAD at the complex number 1, and the electron flow start from complex 1. Then we can also write if we treat with succinate, FAD reduce of FAD increase, reduce FAD, convert into oxidized FAD and provide electron. This electron transport in the electron transport chain. So individual 1, cytochrome C complex, from cytochrome C complex, reduced TMPD doesn't increase oxygen consumption. That means defect is in cytochrome 3 complex or complex 4. Even these complexes 1, 2, 3, all complexes are working. When these things are moving forward, when it comes to cytochrome C, stopped. So likewise, we have to write the answer for the rest of the things. So we just say pyruvate and malate produce NAD, act on complex 1, succinate act on complex 2, reduce TMPD act on cytochrome C. After that, uh, individual 1 defect is on in or after cytochrome C. Individual 2 defect is before complex 1. Individual 3 defect is in complex 1. Individual 1 defect in or after cytochrome C. So you have to again write the second part, exact defect and which complex you found this defect to get the full marks. Okay, then we can